Antigen antibody reaction. An antigen or antibody reaction or antigen antibody interaction. This is a particular chemical interaction between antibodies generated by the B cells of the white blood cell and antigen during the immune reaction. The process of agglutinin combines the antigen and the antibodies. It is a basic biological process that the body uses to defend itself against the various foreign particles like viruses and the toxic chemicals. An antigen antibody complex is formed in the blood when antibodies specifically and strongly bind to the antigen. The immunological complex is a subsequent transfer to the cellular systems where it can be eliminated or deactivated. Antigen. These are the immunogens or any foreign substances that, once entering our bodies, frequently cause a sequence of immunological responses, while others, known as haptens, requires the assistance of other molecules to activate an immunological response. All of the immunogens and the haptens are referred to as the antigen. They could be polysaccharides, lipids, proteins, or the peptides. An epitope is an antibody binding location. Antibody. This is a component that the immune system produces in response to antigen. Thus, antigen result in the production of antibodies. They act together to exhibit the immunological response. The general characteristics of antibody are as follows. An antibody is also known as immunoglobulin. They are Y-shaped, like a protein, generated by plasma B cell. Paratype is the name of the antigen binding site. 5 types: IgG, IgA, IgM, IgE, and IgD. Antigen antibody reaction. The antigen and the antibodies combine specifically with each other. Antigen antibody reaction is the term used to describe the interaction between them. This is a common acronym for it. They serve as the building blocks of humoral or the antibody mediated immunity. This reaction serves as a foundation for the detection of both specific and the non-specific agents, such as enzymes that cause the non-specific disease. Serological reactions are referred to as antigen antibody reaction when they occur in the vitro. Stages of antigen antibody reaction. There are 3 stages to the interaction between the antigen and the antibody. The first stage of the reaction entails the formation of the antigen and the antibody complex. The second stage results in the visible phenomena like agglutination, precipitation, etc. The third stage involves the destruction of antigen or the neutralization of the antigen. Properties of antigen antibody reaction. Significantly specific reaction occurs in the noticeable manner. They are non-covalent reactions like ionic bonds, van der Waals forces, hydrophobic interactions, and the hydrogen bonds. Antibodies and the antigens are not denaturated. They are reversible. The affinity. This refers to how strongly an antigen binds to the certain antigen binding site on an antibody. Avidity. It is the more general concept than the affinity. It represents the antigen antibody complex total strength. It depends on the antibody's affinity. The antibody and the antigen valencies, the number of the binding sites, how epitopes and paratopes are structurally arranged. Cross reactivity. This term describes an antibody's capacity to bind to the similar epitopes on the other antigen. Here we can look for the types of antigen antibody reactions. One is precipitation, next is agglutination, complement, fixation, and the immunofluorescence, and the last one is ELISA, which is enzyme linked immunosorbent assay. Precipitation reaction. An insoluble precipitate of antigen antibody complex is produced when a soluble antigen and the antibody combine in the presence of electrolyte at the specific pH and the temperature. Precipitin is the AB that causes the precipitation, and the reaction is referred to as precipitation reaction. They occur in both liquid and the gel media. Liquid precipitation. An antigen antibody reaction is carried out by adding increasing amount of antigen to tubes containing the constant amount of antibody. Precipitation results from the combined reaction of the antigen and antibody. Gel precipitation. The Petri plates or the plates with the agar gel or a similar gel are used in these methods. In the gel system, both antigen and the antibody rapidly diffuse in all directions. A zone of equivalency observed as a visible precipitation will form at a specific point depending on the diffusion rate and the concentration of reactants. Multiple bands develop in the complex antigen or the antibody preparation. They fall into two methods like single diffusion and the double diffusion. Agglutination reaction. The particles are clustered or agglutinated when a certain antigen is combined with its antibody in the presence of electrolyte at an appropriate temperature and the pH. The clumps of cellular antigen formed by the serum antibody are known as agglutinins. Agglutinogens are the name for the aggregated particular antigen. Slide agglutination. This is a fast and the convenient way to identify the presence of antibodies. Tube agglutination. This is a common technique for estimating the quantity of the antibody. A constant volume of antigen suspension is introduced after serially diluting the AB containing the serum with saline in the multiple small test tubes. A control tube is retained that contains no antiserum. The tubes are incubated by up and until observable agglutination. The tube demonstrating the highest agglutination is known as stretcher. Passive agglutination test. The test is equivalent to the agglutination test, but the physical characteristics of the reaction are different. A carrier particle has the antigen coated on its surface, making the reaction more sensitive by assisting in the transformation of a precipitation process into an agglutination reaction. RBC, latex particles, or the bentonite can be used as a carrier particles. Sometimes tanned RBC, polystyrene coated RBC might be used. Complement fixation. Some non-specific unstable fresh serum components known as the complements are required for the lysis of RBC or the microorganism. Every person has 11 protons that comprise the complement system. They attach to the Fc subunit of AB in the antigen-antibody complex. Complement fixation tests make the use of antigen and antibody complex capacity to fix the complement. In the first step, the test antigen and the antiserum, which have been heated to 56 degrees Celsius to inactivate the complement, are combined with the known quantity of the complement. This is incubated for 18 hours at 4 degrees Celsius. If the serum contains AB that is specific for AG, and AG-AB complex will develop and fix the complement. Immunofluorescence. Fluorescence is the ability to observe the light rays of certain wavelength and emit the light rays of the different wavelength. The fluorescent dyes emit the intense visible light when exposed to UV radiation. In 1942, Albert Burns and the co-workers demonstrated how labeled dyes could be coupled to the antibodies, allowing for the detection of antigen after using the labeled dyes. Commonly used dyes include the most used dye labeled for the immunofluorescence operation is fluorescein, an organic dye that absorbs the blue light and emits a strong yellow-green fluorescence. As a strong emitter of the red fluorescence and an effective light absorber, 30 times more effective than the fluorescein. Phycoerythrin, it is often used as a label for the immunofluorescence. ELISA, enzyme-linked immunosorbent assay. In 1971, the enzyme-linked antigen and the antibody were created as a serological reagent for the testing of the antibody and the antigen. When compared to the radioimmunoassay, they are more simple, sensitive, affordable, and risk-free. The ligand used is a molecule that covalently attached to the enzyme like peroxidases, beta-galactosidase, alkaline phosphatase, etc., and is capable of detecting the antibody. There are three types of ELISA. Indirect ELISA. HIV can be detected using the indirect ELISA method. The surface of the microdata plate is then coated with the envelope proteins developed using the recombinant technology. Unbound proteins are washed out when suspect the serum is added. Sandwich ELISA. This method is used for determining whether there is AG in a sample. The suspect serum is added and given time to react after the well has been coated with AG-specific antibody. Unbound AG is removed from the wells through washing. The next step is to add a labeled antibody against the different antigen epitope. Washing is used to remove unbound antibody, followed by the addition of colored substrate and the color development. The color intensity is direct proportional to the antigen concentration in the serum. Competitive ELISA. This is another variant for estimating the antigen concentration. In this method, the antibody is initially incubated in the solution with the sample containing the antigen. An antigen-coated microtiter well is then filled with the antigen-antibody mixture. The free antibody available to attach to the antigen-coated well will reduce when more antigen is present in the sample. In an indirect ELISA, the addition of a secondary antibody coated with an enzyme specific for the primary antibody's isotype can be utilized to estimate how much primary antibody is attached to each well. Thus, we conclude the discussion with a brief overview of the antigen-antibody reaction. Specific interaction between the antibody and the antigen result in the combination known as the antigen-antibody reaction or the antigen-antibody interactions. Thank you.